Alright, today we're going to go over some basic chemistry and we're going to look at the four types of macromolecules as a review, just as a foundation for later topics. So let's start with carbon. Carbon is the building block of life. It's in literally every organism and in a lot of different molecules as well. It can bond with many other atoms and itself to form complex structures. A good thing to know is that it forms four equal bonds always. So since carbon is everywhere, chemists often abbreviate it by using a different form of representation visually that you'll see on the next slide. So this is something that we'll have to get used to. So on the top here, you have your classic Lewis structure that shows all the different atoms. But now we're going to use something called bond line formula, or bond line notation. So in bond line, each of the vertices here represents a different carbon atom. So for this, we have four carbon atoms, and you can see on the bottom, I've drawn them out here. So we have C, 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 and then we have, remember, carbon makes four bonds, so we fill in the rest of the hydrogens. So 1, 2, 3, 4 bonds. So the hydrogens are implicit in this drawing, and they're not shown. So only the carbon skeleton structure thing is shown here. And this notation basically simplifies molecules so that we can draw them a lot easier and a lot faster. Because it will get pretty cluttered up, like the drawing will get pretty cluttered up if we have all these atoms here. Alright, so we can connect carbons to different atoms that aren't carbon, which are called heteroatoms as well. And then there's a lot of different things we can do. We can form rings, double bonds, triple bonds sometimes. So I just wanted to show all the different types of molecules that you can form here using the bond line formula, the bond line notation. And basically, as you can see, all the hydrogens here aren't drawn in, but they do exist. It makes it a lot simpler to draw. Yep. Then here's some practice. Try to identify how many carbons are within these structures, and also how many hydrogens there are as well. So we'll see this notation continuously from now on, so we'll have to get used to this way of drawing the molecules. All right, so let's go over some basic reactions that we'll see everywhere in biology. So the first one is called dehydration synthesis, and it's also called a condensation reaction. So this is the process by which two molecules will bond together, and water will be removed in the process. So if we see on the right here, we have two glucose molecules. We'll see that in the middle, when we're trying to connect these together, we take an H from one side and then an OH from the other side. That'll make H2O, right? So we'll take that H2O out, and then that'll make a bond between these two molecules. And this is one of the most common types of reactions that we'll see from now on. And like I said, it requires an OH on one side and an H on the other side to come together and form water. Okay, so hydrolysis, this is the exact opposite of what we just talked about. So what happens here is that H2O will come between the two molecules where they're bonded, and it'll separate itself out into an OH and an H, which will break the bond between the two molecules. So as you can imagine, these two reactions will happen continuously pretty much in every type of molecule that we'll look at. So yes, let's keep going. So building off that, we'll talk about monomers. You've probably heard of all these terms, but let's just review. So monomers, these are molecules that they can be connected in long chains in order to form larger molecule structures. So you have a monomer here. Polymers are just the result of those long chains. So a bunch of monomers repeated attached to each other is a polymer. Polymerization, this is just a name for the process by which all these different monomers will link up together to form longer polymers. And a lot of the time we'll use dehydration synthesis that we talked about in order to form these. So each time a linkage is made, we'll lose a water molecule like this. And another term is oligomer. So oligomer just means a relatively small polymer. So a polymer that's only made up of a couple monomers. So you might see that term as well. Okay, so let's go on to our first macromolecule, in our body. Macromolecule just means relatively big molecule that has some kind of important function in the body. So the first type, we'll talk about carbohydrates. So carbohydrates, you can literally just think of these as the sugars, just sugars. So yeah, the monomer of a carbohydrate is a monosaccharide. So several monosaccharides will come together to form a larger carbohydrate. There's also disaccharides, which are when we have two monosaccharides together. Then polysaccharides, where we have 3 or more sugars in a row. So pretty intuitive there. These can have linear structures or ring structures, but often the sugars we'll look at will all be in these ring structures. So on the right side, you see glucose and fructose. You can see they have these hexagonal shapes here, and recall that each one of these intersections is a carbon atom here, and then, oh, that should be just one hydrogen, but you get the point. So we'll have things, hydrogens attached off of them that aren't drawn in, and then same thing goes for this one. And then for naming, often if you see OSE at the end of a molecule, then you can assume that it's a sugar. So I've seen other examples here like lactose, glucose, amylose, arabinose, etc. So yes, all right. So let's continue. So let's look at the function of carbohydrates. The first type is called structural polysaccharides. As you can imagine, they're used for structural functions in the cell. So they can provide reinforcement, protection. So examples of these, chitin. Chitin. This is made up of carbohydrates, and it's in insect exoskeletons. So that hard shell on the outside of insects, that's made of sugar, and it's also in the cell walls of fungi. So it has that protective role. Then we talked about peptidoglycan in our previous lectures. And if you recall, this is in the cell wall of bacteria, and it's made up of all these repeating sugar units right here that provide rigid structure to the outside of the cell. And cellulose, similar concept, but just in plant cells. So this is just a bunch of repeating sugars that provide that hard cell wall on the outside of plant cells. Okay, and then we have storage polysaccharides. These ones are sugars that contain energy. So in our bodies, we have glycogen, which is basically many glucose molecules all bonded together in this branched formation. And we store this inside our muscles so that when we need to use our muscles, we can break these sugars down to get some energy from that. And another example is starch. So starch is the same as glycogen, but just in plants. So plants have that same glucose storage built up so that they can use it when they need. Okay, now let's move on to proteins. So proteins have a wide variety of functions in the body, and they're also the monomers. Like the smallest subunit of a protein is an amino acid. And we'll look at what that means later. So I just wrote down a few functions out of the many. So we have signaling. For example, we have receptors at the surface of a cell that will receive a signal and then send some signal down into the cell. We have catalysis. This just means any chemical reaction that's being sped up by a protein. And we call those, the proteins that speed up reactions, enzymes. You've probably heard that term. Then we have structure. For example, we have the proteins in the cytoskeleton. So if you remember the microtubules, right, that separate the chromosomes during mitosis, that's an example of a structural protein. Then we also have, like, for example, keratin in our hair, our nails, etc. We also have identification, so antigens. And antigens, these are just certain proteins that our immune system can detect and then trigger an immune response based off of that. And then, yeah, many more. Those are just a couple. Yeah, and then for nomenclature, short proteins can also be called peptides or polypeptides. Okay, so let's look specifically at the amino acids that make up proteins. So we have 20 amino acids. There's 2 others technically that exist rarely in nature, but the main ones are these 20. And they have a wide range of properties that are defined by what's called their R group. So if we look at the structure of an amino acid here, we have this central carbon to which 4 things are attached, right? So one is called the amino group, one is the carboxyl group, and you don't have to know exactly what these do for now. So let's just move on, and then we have just a hydrogen. And the most important part is this, what's called the R group, or the side chain. So this R group is the thing that defines every amino acid. So all amino acids have this component in common, and then the R group is the thing that's different. So the R group can have many properties. It can be positively charged, negatively charged, hydrophobic, hydrophilic, etc. It could be really big, it could be really short. So these things will give properties to each amino acid. And the arrangement of these 20 amino acids defines the shape of a certain protein and its function as well. So yeah, here's just some examples of different amino acids. So you can see they have that same backbone structure in common, right? Oh, I should be circling this as well. But then the different part is this R group that comes off. And you can see there's a bunch of different types of R groups here. Okay, so let's look at the 4 levels of how we can define protein structure now. So first we have primary structure. So this is the literal, just the linear order of amino acids. So if we look at a protein, we can see like, we just go in a line and then read off what amino acids there are. So I just put, for example, arginine, aspartic acid, glutamate, etc. So we just go down the line. So we have all these amino acids, and this can be up to thousands of them, and we just look at each one and see the order. So that's the first level of structure that we can look at for a protein. Now let's look at secondary structure. So secondary structure, we're broadening our scope, we're zooming out a little bit. Now we're looking at the chemical interactions that form between the backbones of amino acids nearby. So, and just to refresh, the backbone is this green highlighted part here. And these guys, when all of these are arranged in a row, they can interact with each other to form interesting shapes. So if we look on the right, we can see primary structure is the order here. Then secondary structure, we get these unique shapes that result from the interactions of the backbones here. So you can see we have one's called an alpha helix and one's called a beta sheet here. So you don't have to know the difference between these two, just know that some geometric shapes can be formed from the secondary structure. Okay, now let's zoom out even further. Now we're looking at tertiary structure. So this one is going to be caused by the interactions between the different R groups on all these amino acids. So if you can imagine, all of these have some R group sticking off of them like this, right? And let's say if this is positive, this is negative, these guys will try to get closer to each other. And if this is, for example, polar, and this is polar, then these guys will try to get close to each other. So you can imagine once we have these helices and sheets, all the R groups here, all the R groups will start attracting or repelling each other, and then bend the entire protein into these weird shapes like this, as you can see. So that's what we call tertiary structure, and remember that it's caused by the R groups. So I have, example, positive-negative attraction, polar-polar attraction. You could have polar, non-polar repulsion, and etc., etc. So then finally we go to quaternary structure, and this one's caused when we have, if you look at the tertiary, we're zooming out even further. Let's say we have one of these, we have another one of these, we have another one of these, and then they all come together to form one massive protein. So that's that's what we'll call quaternary structure, when there's multiple protein units that come together to form a larger complex. So you can see there's 4 repeating protein units here, like this. And the classic example of this is a hemoglobin. So hemoglobin it has these 4 subunits, and then each of them will be able to bind oxygen atoms. And the reason this is good is because hemoglobin is the protein that carries the oxygen in our blood, right? So it assembles together, and then it can bind more oxygen atoms, and then it'll carry this through their blood and then deliver it to our cells. And then G protein, you can look this up if you're curious. This is another example of protein with quaternary structure. All right, and then we have some terminology that you should remember or become familiar with. First, we have denaturation. So this is just referring to when we expose proteins to some kind of environmental factor, like heat, radiation, pH, salinity or salt concentration, etc. So what that'll do is that all those, if we go back up here, all these chemical bonds that are forming between these amino acids, right, all along here, etc., all these chemical bonds, if we add a bunch of heat, for example, that's probably going to break some of these bonds, right? And then that'll completely change the structure of the protein that we already have here. So that's what denaturation is, pretty much. Oh, and denaturation, it always refers to some change in the shape of the protein, because the shape of the protein is what determines its function. So if the shape is messed up, then that protein won't be able to function properly. Okay, next we have the substrate. So the substrate is the thing which, if you remember, an enzyme, it's a protein that aids a chemical reaction, and essentially, I don't know what's happening here. Oops. Okay. Right. So the substrate is the thing that the enzyme or the protein that does this will actually do the work on. So the classic example here is lactose and lactase. So if we see, lactose is the sugar in milk, and then lactase is the enzyme that'll break down the lactose. So lactose in this case is the substrate of the lactase enzyme because it's the thing that's being worked on by the enzyme. Oh, and also one thing I forgot to include here is that when you see the ase ending here, pretty much always that's going to be an enzyme. And then if you see this IN ending here, then it'll probably be a structural protein. And if you recall, structural proteins like keratin, tubulin, actin, myosin, etc. These are just proteins that are used to reinforce the cells or reinforce some structure. Okay, finally, the ligand or the ligand. This is the thing that binds to a receptor protein. So we'll see if we have a receptor and it needs to receive some signal. The ligand is this specific molecule that will bind to the receptor and then cause a signal to go inside the cell. All right, let's keep going. So lipids. So lipids, they're a very diverse group of hydrophobic molecules that are relatively nonpolar. So let's look at what that means. If you remember, hydrophobic just means repels water. And if you haven't taken chemistry yet, this nonpolar won't mean that much to you, so it's okay if you don't understand that right now. But eventually you should get to that in chemistry. All right, so lipids, these have 3 main roles. We have insulation, energy storage, and building up membranes. So insulation, just think of like when bears hibernate, they get a lot of fat on their bodies so that they can stay warm. Energy storage, actually I did the other way around, sorry. So insulation is for like walruses because they have a lot of blubber to keep them warm when they're in cold conditions. Energy storage is like the bears getting a lot of food in so that they can hibernate and not have to eat during the winter. And then membranes, if you remember these phospholipids that make up the membrane here, these are another key role of lipids for life. Okay, so let's look at the basic structure of a lipid. So the most common type of lipid is what we call a triglyceride or a glycerol. attached to 3 fatty acids. So we see the glycerol here, we have fatty acid, and then we get this glycerol on the end with these 3 fatty acids attached on here. Okay, and then we can change, the length of these fatty acids or make them, make their composition slightly different, which will affect the properties of the lipid as a whole. So that gives us a lot of options and a lot of diversity for the type of lipids that we can have. And then in a phospholipid, one of these fatty acids is going to be gone, and then it'll get replaced by a phosphate group here, a P. And then if you see this structure, this is where we get this from, because we have the phosphate plus the glycerol here, and then the 2 fatty acids on the bottom. Okay, so these lipids, so we'll briefly go over saturated versus unsaturated. So saturated just means all single bonds in these fatty acids here. So these carbons, they're all single bonded to each other. So I drew that using this bond line formula. Then unsaturated just means that we have double bonds along that chain. So we have carbons that are double bonded to each other. And we don't need to really think about the properties of these yet. It's just good to know that these exist and what these terms mean. Okay, then let's look at what we call sterols. So sterols are a unique type of lipid, and they have these 4 ring structures, as you can see on the right. And remember, each of these intersections are a carbon atom. So we have these 4 rings. Some types of sterols are a lot of different hormones, like let's say estrogen or testosterone or other hormones like that. And then we also have cholesterol, which is a type of sterol. And cholesterol, we'll see, plays a very important role in membranes. So basically, cholesterol will help regulate membrane fluidity. So if you recall the fluid mosaic model of the cell membrane, right? This is saying that the lipids, the phospholipids, are fluid. So it's like we have a sea. It's like we have a sea of phospholipids that can all move around and slosh around, etc. So membrane fluidity, obviously we need some of it so that we can let some things come into the cell. But we also don't want the membrane to be too fluid or too rigid as well. Because if the membrane is super rigid, then nothing's going to be able to come through. No nutrients will be able to come through. And then if it's really fluid, then the cell might just like fall apart because it's like, the phospholipids aren't close enough together. So let's see what cholesterol does to regulate this fluidity. So in low temperatures, since it's getting cold, all these lipids are freezing up, sort of. They're like compacting together, they're getting really tight, so the membrane is getting very rigid. Basically what cholesterol does is it inserts itself between these phospholipids like this in the membrane. And by doing that, when it's too cold, the phospholipids are trying to get really close together and compact and freeze up. But the cholesterol literally physically separates the phospholipids so that they won't get too close together. So that in low temperatures, cholesterol will keep the membrane fluid enough so that it can still function as normal. Then let's look at the opposite. So when we have high temperatures, these phospholipids, they're getting really far apart, the membrane's getting really sloshy, liquidy, and it's like threatening to fall apart, right? So in that case, cholesterol is embedded in the membrane here, and it acts as sort of like a scaffold on which the phospholipids can like build off of. So it actually is like holding the phospholipids together when they're trying to like fall apart. So in high temperatures, cholesterol will maintain the rigidity of the membrane by doing this. So we can see it has this like kind of like dual role in the cell here. All right, so let's move on to nucleic acids. Nucleic acids, these are storing and transferring genetic material for the most part. This is mostly all review, I'm sure. So we'll have two main types. We have deoxyribonucleic acid, DNA, and then ribonucleic acid, which is RNA. And they're differentiated by the sugar that they have. So DNA has deoxyribose, and RNA has ribose, right? Right, so DNA is, everyone knows this. It stores the genetic information. It's usually double-stranded, right? And some other things about it is that it's pretty stable. So it doesn't just fall apart on its own. It stays, it stays in its normal conformation. Then it has ATGC, adenine, thymine, guanine, cytosine. Okay, now let's look at RNA. RNA, it transfers that genetic information. That's one of its roles. It's also usually single-stranded, although we'll see, or I guess we've already seen some exceptions because we looked at viruses that have single-strand RNA genomes. It's also relatively unstable, so it can kind of just fall apart, and it can be easily broken down by enzymes. So it's sort of transient. It comes and goes pretty easily. And then it can also have some other functions in the cell. So there's some rRNA, tRNA, siRNA. If you don't know about these, feel free to search them up. But we'll go over at least the first two in detail when we talk about transcription translation. Right? Also has AUGC instead of ATGC. You can see the structures on the right. Yeah, pretty self-explanatory. So yeah, let's look at the structure of nucleic acids. So the nucleic acids are made up what we call nucleotides here. This is one nucleotide, right? And the nucleotide, it's going to have a sugar. If you remember, the sugars often have this ring shape, right? Then it's also going to have a phosphate group, a P. Then what we call a nitrogenous base. It's just this big organic molecule here that has nitrogens in it. And then the nitrogenous base is like what gives it the letter here, right? So adenine, thymine, guanine, cytosine, or uracil if you're an RNA. And then quickly, let's talk about 5' and 3'. So in DNA and RNA, we have what's called 3' and 5' ends, and this is what helps us orient the strand in the correct direction, and so that we don't flip it on accident and don't know like what order we're looking at the DNA in. So basically, you have, if we look at one nucleotide here, so if you remember, each one of these intersections here represents a carbon. So what we do is that to specify the number of the carbon, we can label going from right to left. So this will be 1, 2, 3, 4, 5. So you can see that when we attach a bunch of nucleotides together, the 3' carbon here always has an OH attached to it, what's called the hydroxyl group. So this is like how we tell like what direction the DNA is oriented in, because on one end it has 3', then on the other end, at the end of the chain, let's erase all of this. So again, we have all these carbons, right? 1', 2', 3', 4', 5'. So on this 5', so it's actually, sorry, it's not drawn in here, but this should have a phosphate attached to the end here. But anyways, yeah, so the 5' end will be the one with the phosphate. So if we look at a DNA sequence, one end will always have a phosphate sticking off because of the nucleotide arrangement here. Then one end will always have this OH sticking off because that's how the nucleotides are arranged. So we can basically specify what order we're looking at the DNA in. So this, if we're looking from top to bottom, we're looking from 5' to 3' end. Okay. And I think before I might have circled this bottom as part of this nucleotide, but that's actually incorrect. So it should be, I'll move to this one. So here, like this should be the nucleotide, if that was unclear. But either way, the components are the same, so it's fine. Okay. Okay, so let's look at, so usually when we talk about nucleic acids, we just talk about DNA and RNA, but there's also, if you've heard of, I mean, you guys all know ATP. ATP is also a type of nucleic acid. Then we also have something called GTP, which is very similar. So these are also nucleic acids, but they're actually used for energy storage. So now you have the sugar and the nitrogenous base, but now you have 3 phosphates on the end, TP, triphosphate. So both of these are used in our cells for energy. So what we do is we'll cut off one of these P's. That'll release some amount of energy that we can use for other processes in the cell. So ATP triphosphate to ADP diphosphate, okay? And then GTP to GDP as well. And then the difference between these two, ATP is kind of used everywhere throughout the cell. It's like the most common form of energy storage, while GTP is used in more specific situations, and specifically those related with the G protein receptor pathways, which you might not learn about. Actually, I think you guys might learn it in AP Bio, but eventually you'll hear about these when you take bio classes in the future. And these are pretty important in our bodies because they're one of the main receptor types for our cells. Okay, now here's just some practice problems. So basically, you can just go back to the previous slides and look at what the structures of each type of macromolecule are, and then try to identify which out of the 4 macromolecules each one of these structures is based off of that. This is good because it's good to be familiar with these bond line drawings, because you'll see these a lot in the future, and when we're talking about other concepts, if I draw something like this, then you need to know that that's a sugar automatically. So yep. So just look over those, and then we'll just go from there.